Very happy to meet you all in this video platform. This is Dr. Gandhimadi, working as professor in the Department of Pharmaceutical Chemistry and Analysis, School of Pharmaceutical Sciences, Wales Institute of Science, Technology and Advanced Studies, Pallavaram, Chennai. Today I am very happy to share some information about mutation. Before going to this topic, let's have an introduction about the DNA. DNA is a double-stranded helix genetic material, have sequence of purine and pyrimidine nucleotide. Among two strands, one strand acts as a sense strand where transcription occurs to form a messenger RNA contain codon. This transcripted codon undergoes translation process to form polypeptide. Coming to this mutation, if you want to define a mutation, that is the change in occurs in that nucleotide sequence of the DNA. So already we have know that DNA is having that sequence of the nucleotide. If there is any changes occurs in the any one of the nucleotide, that is called as DNA mutation. This DNA mutation results in permanent changes in the DNA sequence. So if the mutation occurs in the germ cells, if that mutated germs transmitted to that human being, it will produce that inherited diseases. Whereas in the mutation in the somatic cells, it does not transmitted any diseases, but it causes the cancer and some malfunction in our human being. Let's have some related terms to that mutation, that is mutagens and mutagenesis. The mutagens, that is nothing but agents which cause the DNA damage or agents which are reason for the mutation, that is called mutagens. Some example for the mutagens are X-ray and UV rays and some chemicals, and the most important one of the chemical is acridine orange. So this rate of mutation is proportional to the dose of irradiation, which means when a person they have more exposure to that mutagens, they will get the more possibilities for this mutation. And the another term related to the mutation is mutagenesis. So mutagenesis is nothing but process where the mutation occurs. So that is called mutagenesis. And this is what are causes for the mutation. That is it is very important to know about the causes of the mutations. The main causes in the causes of mutation is errors occurs during replication. That means when the replication occurs, if there is any mismatch pair is formed during the proofreading, so that will be a major reason for this mutation. And another error due to recombination events, that is also one of the major cause. And third one is spontaneous change in the DNA. That is the usual chemical change occurs, or there is a chemical change or the nucleotide changes. That means the cytosine is one of the amino pyrimidine nucleotide, but this it is easily undergoes the deamination and it is easily converted into the uracil. So instead of the cytosine, that is uracil will be replaced, or that will be occurs. So this is one of the frequent reason or a common reason for the mutation in that DNA. And another one is the purine nucleotide easily undergoes the depurination, and it is also one of the major reason for the mutation. And that environmental factor like chemical mutagens and irradiations like UV light and ionizing radiation, these are the other environmental factor for causes the mutations. Coming to type of mutation, we can classify the mutation majorly into two types. One is point mutation and other one is frame shift mutation. The point mutation we can call as a base mutation. That is means there is a when you when you take a DNA, it have this sequence of the base. In this, if one base is replaced by the another base, that is called point mutation, and another one is frame shift mutation. So in this mutation, what will happen, if one new nucleotide, it is if it is inserted, or one of the nucleotide from the frame or from the DNA is deleted, so what the totally it will changes the sequence of that nucleotide, and the total frame of that, or more the total frame of the DNA get changed. That is the frame shift mutation. The point mutations are further classified into three types: silent mutation, missense mutation, and nonsense mutation. Among the missense mutation is it is maybe acceptable, partially acceptable, and unacceptable. The frame shift mutation further classified into two types. One is deletion frame shift mutation, and another one is insertion frame shift mutation. Depends upon the nature of this and type of the mutation, different diseases is occurs. So first we can I want to explain about that base substitution or point mutation. So in this mutation, in this only one base is get changed, or the changes occurs in the one base in the DNA sequence. That is called point mutation or base mutation. So this is classified into two types. One is transition type or transversion type. In the transition type, this purine is replaced by the another purine, or the change of the nucleotide occurs within the same chemical classification. This, if pyrimidine base, for an example, this cytosine and uracil, this cytosine, uracil, and thymine, these are the pyrimidine bases. If instead of the cytosine, uracil is replaced, or instead of the cytosine, thymine is replaced, so this change is called as transition type. Like that, purine bases in the DNA is adenine and guanine. So if adenine is replaced by the guanine, or guanine is replaced by adenine in the DNA sequence, that is called transition type. In the transversion type, the purine base is replaced by the pyrimidine base, or pyrimidine base is replaced by the purine base. That is called transversion. So this example help you to understand well about that transition mutation and transversion mutation. For an example, CAT, this is one of the sequence in the DNA where the C is changed to, or it is substituted by the T. So we are getting, instead of the CAT in the normal DNA, we are getting the TAT in the mutated DNA. So here C is changed to T. So both are coming under that pyrimidine nucleotide. So this is called as transition mutation. And another example is CAT. That is, in that C is changed to, or replaced, or substituted by the G. C is a cytosine, that is a pyrimidine nucleotide, whereas G is a guanine, it is a purine nucleotide. Here the purine nucleotide is changed or replaced by the, that is a purine nucleotide. So this is one of the example for transversion mutation. If you want to explain in that messenger RNA, mRNA or messenger RNA wise, or in the frame wise, see, this is a sequence. AUG is a one codon, and the second codon is GCC, and UCA is a third codon, and the fourth UGC. Like that, the codon sequence are there, and their respective, each codon is responsible for the particular amino acid. AUG is a start codon which is responsible for methionine amino acid. GCC is a alanine and UCA. So this is a normal messenger RNA, and when it undergo there is a transaction, it will produce a normal polypeptide contains the sequence of methionine, alanine, serine, like that polypeptide we will get it. If for an let's if an example, in that third codon, the second base in that normal messenger RNA is C, if it is substituted by that U, we are getting UUU is a third codon in that mutated messenger RNA, which is producing the phenylalanine. So we are getting that nucleotide where there is in the mutated or in that base changed codon producing that different amino acid. So we are getting a polypeptide in that which sequence is mutated. In that particular sequence we are getting a different amino acid. So this is the example for that transition type and transversion type mutation. And this point mutation produce it may be a silent mutation, missense mutation and nonsense mutation. This is in that silent mutation, we are not observing any there is a detectable effect or any harmful effect we are not observing in that silent mutation. How it is not harmful means there is in that codon synonym codon is there. What is synonym codon? The synonym codon is nothing but more than one codon which is responsible for the same amino acid. I just clear you, and that CGA is one of the codon which is responsible for the arginine, and another codon CGG, this is also responsible for the arginine. So in a sequence where CGA undergoes mutation and in that the A is changed to or it is replaced by the G, we are getting the CGG codon which is also producing the arginine. So which results in there is no change in the amino acid sequence in the polypeptide. So we are not getting any harmful effect. So this is a silent mutation. And another mutation is missense mutation. So in this missense mutation, we are getting the different amino acid, because in that when a codon undergoes that in a particular codon in the messenger RNA undergoes that mutation where one of the base is replaced by the another base which is producing the another amino acid. So we are getting a polypeptide where the particular codon where it the mutation occurs, that particular codon will produce a different amino acid. So based upon this location of the mistaken amino acid in the specific protein, missense mutation may be acceptable, partially acceptable or unacceptable with this respect of the function of the protein. Coming to acceptable missense mutation, for an example, in the hemoglobin Hikari. So here in the normal hemoglobin, in that 61 position of that hemoglobin, in the usually lysine is we have a codon AAA or AAG which is responsible for lysine. Whereas in the hemoglobin Hikari, due to missense mutation, in the AA and the third base A is replaced by the U or the G is replaced by the C. So which producing instead of the lysine, it producing asparagine in that 61 position of the hemoglobin. So this is not produce very harmful, but it is acceptable one. In the partially acceptable missense mutation, that is a sickle hemoglobin. Here this is a in the normal the amino acid in that position 6 is beta chain of the 6 in hemoglobin is there is a glutamic acid. during mutation, that is the codon which is responsible for the glutamic acid in that sixth position of the hemoglobin, it may be GGA or GAG in that normal hemoglobin. If it undergo mutation, this second position, that is the second base of that GGA, is replaced by the U which produce GUA, or the A is replaced by the U which producing the valine. So instead of the glutamic acid, the valine will be present in the sixth position of the beta chain of the hemoglobin. This is producing the sickle anemia. And the third one is unacceptable missense mutation. In the unacceptable missense mutation, it is methemoglobin. This disease is by unacceptable missense mutation, in which the normal amino acid in the position of 58th position of the hemoglobin is histidine. Alba chain of the hemoglobin is histidine. It is during a mutation, the codon which is responsible for that histidine is CAU or CAC in the 58th position. When the first base of the cytosine is replaced by the uracil which produce the UAU or UAC, which is producing the tyrosine. So in the place of histidine, tyrosine amino acid is replaced, which produce the methemoglobin. That is a non-functional protein which cannot transport the oxygen. It is a very dangerous one. So this is called unacceptable missense mutation. Coming to nonsense mutation, which leads to the conversion of the amino acid codon into a stop or nonsense codon. So everyone knows that stop or nonsense codon is, these are the codons which does not code for any amino acid. So when this during that protein synthesis, when the stop codon or nonsense codon reaches, it will with that the polypeptide synthesis will be stopped. So here in that this is one of the best disease by nonsense mutation is thalassemia. In the thalassemia, the 17th codon of the beta chain that is in the normal DNA, in the 17th codon of the beta chain is UGG, which is responsible for the tryptophan amino acid. When it is mutated in the third nucleotide, G is mutated into A, it produces the UGA. So it is a stop codon. So because of the stop codon, this up to 17th position only the amino acid will be synthesized, and after that there is no amino acid will be synthesized, and no formation of the or lengthening of the polypeptide. When this stop codon reaches, with that the nucleotide or polypeptide formation will be stopped. So we are getting the premature polypeptide we used to get it, which is non-functional. And this frame shift mutation. In the frame shift mutation, this is occurs when one or more base is externally inserted in, or one or more base is deleted in the base sequence of the DNA. So what will happen? The total base sequence of the DNA strand get changed. So this is called frame shift mutation. So this is a difference between the point mutation and frame shift mutation. In the point mutation, the particular codon which is amino acid, that particular amino acid will be changed, whereas in the frame shift mutation, this one where the in which codon the frame shift mutation is started, from that codon onwards, the remaining all sequence of the amino acid and everything will be changed. That is, the frame shift mutation by insertion is explained with this diagram. So in the let's have an example. In the normal DNA, we have a sequence of AUG, the codon sequence GCC, and their respective amino acid sequence are given in this. See, if in the third codon UCU, if C is inserted, so what will happen? The C will join with that the codon, the first two base of the UC. So UC will be formed, and this the third base of that third codon of the normal DNA will be moved to that the fourth codon. So there it will form a UUC, and that the third base of that fourth codon will be joined with that, or it will be linked with that another codon. Like that, the one during the insertion, the last base of the each codon will be joined with that the another codon. Like that, the total frame get the codon will be changed in this mutated messenger RNA. See, in that because of the insertion in this messenger, that is a mutated messenger RNA, we are getting where the mutation occurs. From that, the total amino acid sequence get changed. So that is the insertion. And another example, frame shift mutation by deletion. Here you just see that is the one of the in this normal. This is the first one is the normal messenger RNA, where this having the sequence of the codon AUG, GCC, UCC, like that, which is and their corresponding or respective there is a amino acid sequence are given below. During that deletion in that third codon, there the actually in the normal messenger RNA we have the CUC is a third codon. Here the first base U is deleted. So what will happen? The second base CU will pair with it will form a codon with that the first base of that fourth codon, and that the remaining two codon of the fourth will join with that first codon of that fifth codon. Like that, the codon will be changed. So where the mutation occurs or deletion occurs, from that onwards the amino acid sequence will be changed, and we are getting the entirely different protein than the normal protein. So with this notes, I just conclude that mutation.